So we are going to start a new chapter today. So in the last class, what we have done is we have completed the first chapter, that is reproduction in organisms. The next chapter that we are going to start is strategies for enhancement of food production, right? So in this chapter, what is most important from entrance point of view? The most important thing is the chart that is given in your NCERT book. If you have gone through the chapter or if you have looked into it, what you will see is there is a chart called as chart 9.1 and 9.2. So the most important point or most important part of this entire lesson is the two charts, right? So maximum questions comes from that two chart. Have you seen the chart? There are two charts. In one chart it is given resistance against disease, and in the second chart that is something called as resistance against pest. So in this entire chapter, the most important part is those two. Apart from that, what we are going to deal in this chapter are, or what are the things that this chapter contains. Here we have to talk about animal husbandry. Sorry. Right. So what we are writing is the contents. Right. Kiki porhim, animal husbandry. Next we'll have plant breeding. Third is green revolution. Fourth is single cell protein. Fifth, we'll be talking about green manure. Sixth is about energy crops. And last, we'll be talking about tissue culture. Right? So we should have an idea of what are the contents of this lesson, right? So these are the contents. Out of these points, animal husbandry is something that will be done in your zoology class, and in botany what we are going to discuss is from point number 2 to point number 7. So we'll be starting with plant breeding, right? Animal husbandry, zoology korabo. So that will be our first topic. Before going into details on this topic, let's talk about the name of the topic, right? Or name of the chapter. What is the name? Strategies for enhancement of food production, right? So we are going to discuss certain methods which are involved or which are applied for increasing the production of food. Now, why is it necessary that we have to increase the amount of food, right? Jimankini food etia ase, is it not sufficient? So what we know is, like, about amongst all the problems that this world is facing, right? Ajiloke jimanbur problems world khone dekha paise. Tar bitor the most important problem that it is facing is obviously shortage of food, right? So as far as the comparison between population and food is concerned, so population is always increasing, and tar in relation ki hoise food or content to iman besi bahra nai. So because of shortage of food, science has come out with various types of methods so that they can solve this problem. And what are the methods we'll be talking about is, we'll be talking about all these methods that we have already written. For example, animal husbandry, plant breeding, tissue culture, right? So, ebilak methods apply kori palai, we are trying to solve that shortage of food. So, this is what we have to learn, right? So, the first topic is, and another thing is that you have to know at the first beginning is, out of all the plants, right? Jimanbur, whenever we talk about food plants, no, generally food buli kole, what is that thing that comes to our mind is something rice and wheat, most staple food, no? Eta part of wheat khai, eta part of rice khai. There are so many other foods also, but rice and wheat are the staple food. So maximum of the varieties of rice and wheat that we found it today are all the products of plant breeding, right? So increase tar contents bilak increase koribo lagile ki korise scientifically various varieties are ulaise, various bhal traits bilak ulaise, improved varieties, improved yield. So this is what is called as the process of plant breeding. Now, what is plant breeding? Plant breeding ki bostu? Plant breeding is nothing but it is an art and science of changing the genetic makeup of a plant to obtain desired characters, right? So plant breeding ki hoy, etu eta method, right? So etu artificially kora hoy, or plant breeding ami kiho karne koru, or what do you do? Plant breeding, to understand it in a very simpler method, what we do, je ami duta parent choose koru, ar duita parent or jun bilak desirable characters thake, those characters we try to incorporate it into one plant, right? Progeny to ami jate ami bisara traits bilak thake. So what we do is we make a cross between that two, and that process is called as plant breeding. Why do you go for plant breeding? We go for plant breeding because we want the product, or we want the result according to our wish. For example, if we have two plants, adult plant or ki dekhisu, adult plant, if you are talking about, maybe we are talking about two rice plant, right? Adult rice plant or ki dekhi, so the grains are longer and the quality is good. And aru adult kintu, suppose, okay, we'll give the example. Suppose we are talking about rice plant 1 and rice plant 2, right? In this rice plant, tar eta nijor characteristics thakibo. For example, it has long grain, right? Aru it has long grain, aru it takes more time to grow, right? Our rice 2 বা second plant dalot, it has small grains and it takes less time to grow, right? Less time to grow. Just a very simple example. So what we want is adult plant oru kisuman advantage ase aru adult plant oru kisuman advantage ase. What is the advantage? That the grains are long, and we know whenever the grains of the rice are long, then obviously the rice that we'll eat, that will be it will be very tasty. It might be very tasty, and but as far as the look is concerned, the look would be very beautiful, right? So if you are talking about the beauty of the rice, it comes from this plant, but the disadvantage of this plant is it takes a longer time to grow, right? But in this case, the grains might be smaller, but it takes a lesser time to grow. Now, what we want is, we want a such type of a rice which has both the characteristics, right? So, amak enuka dal rice lage, jot niki yare bhal characteristic to, or yare bhal characteristic duita combine hoi thake. That is what is called as plant breeding. What we will do, we will make a cross between this plant and this plant, or tar pisot, tar juntu progeny hoy. That progeny, after evaluation, if that comes out to be the best rice with all the best qualities according to what we desire, that process is called as plant breeding. Or plant breeding or karone ami ki pao, we get a plant with the desired characteristics. Buji paiso? Hello? Okay. So that is the reason why we go for plant breeding, right? So maximum of the crops, maximum of the crops, right? Or many crops or many examples are there which are results of plant breeding. Certain examples that we can write is, if you are talking about rice, there are so many varieties of rice. For example, we have basmati, right? Another rice variety could be Annapurna, right? There are so many other varieties. Similarly, in case of wheat also, if you talk about, there might be some varieties called as Kalyan Sona, right? Next is Sonalika, right? Secondly, if you are talking about some other crops like castor at a variety, Aruna variety of castor, etc., etc. So these are certain varieties which has been developed because of plant breeding. So normal rice plant asile, normal wheat plant asile. What we have done is we have made a cross with some other with other plants. With other plants, obviously kundal plant or lokot koriba, the plant that has the desired characteristic, right? And ultimately we produce some new varieties, and this process is called as plant breeding. Right? So what we understand is plant breeding or karone, juntu progeny that is formed, that new variety will always be with the best characters that we have desired. Next is, once we have developed a new variety of rice with all the characteristics that we want, ami keneke gom pam that plant is improved. Plant breeding or main kam ki? to develop a improved variety, right? Next point is we have to know that the plant is improved. So there should be certain characteristics that are seen in the plant that tells us that the plant is improved as compared to the previous, right, or as compared to the parents. So how do you know that the plant is improved or plant improved puli kobo lagile kisuman characteristics thakibo no, and what are the characteristics? Adult plant ok ami improve buli ketia kom ba better buli ketia kom? Are you understanding my question? So there should be certain characteristics on the basis of which you will consider the plant to be improved, right? Can you suggest any characteristic which qualifies a plant to be improved? Adult plant ok improve ketia kom? Yeah, it should have a better crop yield, right? The quality should be good, the quantity should be good, right? It should be like the yield should be good. Then it should be resistant also. We should talk about this factor. Resistant to what? Resistance to pest, right? And third, we could talk about tolerance. Freeze है? Okay. Okay, chalo. So we were talking about plant breeding. What is the most important function of plant breeding, or why we go for plant breeding? So that we can get an improved variety of a crop, right? How can you say that the plant has been improved? By looking at certain points like this. The new plant that we have made should give better crop yield, no? Production to besi hobo laibo. Next is it should be resistant against pest, and thirdly it should be tolerant. Tolerant against what? Tolerant against environmental conditions, right? Jikunu environmental condition of jodi grow koribo pare, then what we say is that plant is tolerant for any adverse environmental conditions. So if a new crop shows all these characteristics, we can conclude that the plant is of improved variety. Or improved variety keneke banao? Obviously plant breeding or through banao, right? Now how do you go for plant breeding, or how do you do plant breeding, right? There are certain steps for plant breeding. Steps for plant breeding, right? What are the steps? First is collection of variability. Second, evaluation and selection of parents. Third, selection and testing for superior recombinants. And finally, after doing all this, we go for testing, releasing and commercialization. Right? So in order to go for plant breeding, these are the things that we do. Plant breeding or ki hoy notun plant etal produce koru, kiho karone produce kore, so that we get a better quality or we get better taste, right? And the new plant that we have made, code bonaisu, obviously in the lab or in that field area that we have produced, hey bostu to ultimately ki koribo laibo? We have to commercialize it. So in order to begin with step breeding, the first point is something called as collection of variability. What we understand by collection of variability, tamane jikunu eta bostur upor jodi if you want to study, for example, if you are talking about mango or if you are talking about rice, ki hol say you want to study about the rice plant or you want to study about the mango plant. So what would be your first step? If suppose your study is based in India, tetia hale tat ki koriba say India bhitor jiman bor rice grow kore ba jiman bor mango grow kore no, jiman bor beleg varieties ase ei guti mango bor ba guti rice bilak tumi collect koriba, right? After collecting only, then you will go for the further studies. So the first point of plant breeding or the first step of plant breeding is always collection, right? So beleg jagat jot beleg grow korisa. For example, if you are talking about rice or you are talking about mango ami ki jano, there are so many varieties, no? Alfonso ase, Ratnagiri ase, right? Darshini ase. So kiman beleg dhoroner mangoes ase, plus there is always one particular mango that is common or local to that particular area. So if you want to study any particular plant, first thing that you will do is always collect. And so the first step of plant breeding is always collection of variability. After collection, tar pisot ki korim? Je ami best to select korim. Best to select korar pisot, we'll make a cross among the best that we have selected. After that, after the parents has been selected, tahator major cross kora pisot junto offspring hole, that is something called as the progeny. Jimanbur offsprings hole, tar bitor junto superior hoy, that is something that we will select. After that superior variety has been selected, it will be tested again. Our testing kenege korim no, adult plant ami test kenege koru that we keep on growing that plant for many growing seasons, right? So bahut bikita seasons of grow kori ultimately junto best variety, that variety goes for release and commercialization. So ultimately someone will have to say that out of jimanbur experiments ami korisu, the plant that we have made is now ready for commercialization or it's ready to be sent out, right? Or it should be, or kunuba ektu kobo laibo that this is the best. Kune kobo, or we have to talk about something called as certification. Jeneke porikhat, you keep on writing and there is a teacher to tell you whether that the answers that you have written is right or wrong, right? Tomak eta certification ba eta certify kunbai koribo tahibo. Thik teneke iman khini korar pisot jundal plant ami produce koriso, hey plant dal je bhal, but it is proper or it is best, someone needs to certify. And who is that someone we are talking about? Ami all of pisot likhim. This is nothing but National Seed Technology ba National Seed Bank bilak jun bilak thake, tahare ki kore certify kori dia. Ar jetia he seed to best boli koy, that seed is ready to be sown. So we will be coming to the first step, that is collection of variability. Whenever we talk about collection of variability, we have a term that is called as germ plasm. Have you heard that word? Germ plasm kunisila niki? Germ plasm. So we are talking about collection of variability. Right? What is collection of variability? It is the first step for plant breeding. So discussing this, we have something called as germ plasm. What is germ plasm? It is the sum total of all the alleles for all the genes present in the crop. So this is how the definition of germ plasm goes like. But in simple language, germ plasm mane ki? What is germ plasm? Something that is related with, hello? Yeah, so the question is, what is germ plasm? But germ plasm, what strikes in your mind? We are talking about the collection of the genes, right? Jikunu eta crop or various varieties take, are ebilak the different varieties take. Who is responsible for the creation of these varieties is obviously the gene structure, right? Yeah, so adult plant or idal plant, they differ in their gene structure, and as a result there are different varieties. So in order to study a particular plant, what you need to do first is you have to collect all the genes or all the varieties, and in the varieties jimanbur genes ase he gutei genes khini tumi study koriba. That collection of all the alleles of the gene is called as germ plasm. As far as the word allele, genes and all this stuffs are concerned, I think in genetics we have talked about, right? Alleles ki hoy, alternative form of a gene is called as allele, right? So ekdom from entrance point of view, so definition to monot rakhibo nalage, right? So what you need to remember is the word germ plasm. Germ plasm, very short ke, what you understand is collection of all the varieties of genes that are present in that crop. Right? We are not talking about one species or two species. We are talking about the entire species of that particular crop. Right? Next is some information about germ plasm that you have to know. So what is germ plasm? So we have discussed sum total of all the alleles of all the genes is called as germ plasm. One second. Okay, next is from where will you collect? So plant breeding kori aso, plant breeding or first step ki collection. Ki collect korim? Genes bilak collect korim, no? The next point is genes bilak korpra collect koriba, right? So this germ plasm could be collected from either within the country or it could be collected from outside the country, right? Okol je India prai collect korim, not necessary, outside the country, right? So when you collect the germ plasm within the country, that type of a collection is called as indigenous collection. Indigenous collection, or it is also called as IC. And if you collect the germ plasm from outside the country, we have a word that is called as exotic, right? Exotic collection, and that is what is called as EC, right? So out of this, what you need to remember is what is the difference between indigenous and what is the between indigenous and exotic. So India bhitor jedi tumi collection kini korisa, that is called as ki indigenous. Jedi beleg countries of price outside India, detail tak ami ki boli kom exotic. So you have to remember the meaning of exotic. Next is when you have collected the germ plasm, what is the next step? You have to maintain that germ plasm, and while maintaining, we need to remember that it is always maintained at low temperature, right? So what we are saying is prothomote genes kini collect koribo lagibo. Now genes to aru iman dangor bostu nohoy je tumi goi pela hatere collect kori aniba, no? Amak ki lage? Amak beleg varieties. Suppose we are taking mango as an example. Amak varieties of mangoes or amar eta product lage so that we can grow in the next season, right? So for collection purpose, what is the best plant part that we take is always seeds, right? Adult plants, we want to keep it for the next generation or we want to study about it, the best part that we study is always the seed, right? So in case of seed, generally a seeds bilak ami kihot maintain koribo lagibo. If you want to keep it for a longer time, you always have to keep it in low temperature. While we are talking about low temperature, there is another word called as cryopreservation. Hunisane? Have you heard that word, cryopreservation? Okay, if you have not heard cryopreservation, have you heard something called as liquid nitrogen? Yes, so liquid nitrogen, cryopreservation, right, means what? Cryopreservation, preserving in a very low temperature is called as cryopreservation. So seeds bilak jate niki beya hoi na jay, so tahato ki kore generally seed bank bilakot preserve kore, no? Seed bank bilakot one of the process of preserving is through cryopreservation also. Cryopreservation or ki kore is liquid nitrogen or bostubur thoi diye so that the seed does not get destroyed. Iman kini details na lage. As of now, what we know is once we have collected the germ plasm, it has to be maintained. Where will be maintained? We'll maintain it in low temperature. Generally, this statement is in relation with the seeds that we have collected. Next is if the germ plasm we have collected is from fruit trees. So in case of fruit trees, germ plasm is maintained as trees grown in fields. So kisuman okol je ami low temperature ba seed bank ote je seed bilak thoi dim, not necessary. Ketiaba ami ki koru for our study purpose ami kisuman plants o collect koribo paru, no? So jedi ami plants bilak collect korisu for our study, hey plant bilak ami maintain keneke korim? Tahatok to aru liquid nitrogen ba low temperature thoi dile he gosdal to muriye jabo, right? So in that case, if we are talking about trees as our point of study, ami hey tree dal maintain keneke korim je ami he dot kobat belegot plant kori thoi dim. So if we are talking about fruit trees, the germ plasm is maintained as trees. Aru kot a trees bilak thakibo ট্ৰিজ বিলাক যাতে জীয়াই থাকে তাকটো আমি ৰুই পেলাই থব লাগিব ন এনে আনি পেলাই কাটি পেলাই তো গছল থৈ দিলে নহব so in that case we grow them in the fields and by growing them we preserve or we try to maintain the germ plasm of that tree. Is the point understood? Okay, next is the last point that we have to know about germ plasm is why do you have to preserve the germ plasm ba ami germ plasm collect kiyo koribo lage, right? Why is it necessary? Germ plasm collection is necessary and what is the reason behind? Why is it necessary? Necessary collection is necessary because kyo kare some points like agricultural expansion, right? If you want to expand the agricultural system, then you can do it with the help of the germ plasm that we have collected. Second is due to some other human activities. Right. Third could be to go for a suitable breeding program. For conducting suitable breeding program, right, and etc. So these are the informations about the first step, that is collection of variability, what we have discussed. So what we have discussed is plant breeding ami kiho karone koru. We are talking about plant breeding. What is plant breeding? It is a method of what? To increase the crop yield, right, or to get a plant of desired characters. To go for that method, ami kisuman steps follow koribo lagibo, and out of the steps, the first is collection of variability. How will you collect the varieties that are present in that crop? You go for collection of the germs or the alleles, right? And that collection is called as germ plasm. Germ plasm to collect kori pelai tak maintain koribo lagibo. The first point is ami collect korim. It can be either indigenous collection or it can be exotic collection. Next is we have to maintain them. Either we can maintain them in the field or we can maintain them at low temperature in some banks, right? Next is kiho karone collect koribo lage? To solve some problems like we have to talk about agricultural expansion, some other human activities. Other human activities mane ki? Maybe you might be talking about an area jot niki at a particular type of vegetation grow kori asile, right? And in that area what happened is human beings started building up flats there, right? So agricultural land to gusai pelai residential area bonaboli khuzile, hey tat thaka jiman bor species ase hey blak to naikia hoi jabo, so that all those species does not get extinct from the world. What we do is we try to collect the germ plasm for our future need, right? So this is what we understand by other human activities, and obviously we go for germ plasm collection for suitable breeding program. So this was the first step of plant breeding. After collection, what next? Plant breeding or ami collection korilo, collection korar pisot ami ki korim? We have to, now we have to select the parents. Means what? We have taken a bank or maybe a area tat ami ki korilu je ami bahut bostubur we have collected, right? After collection we have to select. What we are selecting is the parents, no? And why we have to select the parents? Ami parents kisuman ba suitable parents select kiyo koribo lage? So that tahator majot cross eta kori we can get a new plant, right? So the second point is selection, evaluation and selection of parents. Evaluation and selection of parents. So what we do here, out of the entire things that we have collected, we will select the parents with desired traits. What is a trait? Trait ki bostu? What is a trait? A trait is nothing but a character, right? So কোনটো parent choose কৰিম আমাৰ main aim কি যে দুটা parentক আমি combine কৰিম কিহৰ কাৰণে combine কৰিম যাতে নেকি আমি দুইটা parentor best quality is something that we obtain in the next generation, right? While choosing the parents we have to be careful and we have to select the parents with the desired trait. Desired trait in the sense ki maybe we are talking about a plant A that has a high protein yield, right? And we are talking about a parent B that has maybe disease resistance, right? So kundal parent or high protein yield ase kundal parent or disease resistance ase hetu bostu ame select kore and this is what is the meaning of the second point, right? And why do you do so? So that we can get a cross and we can get the F1 generation that is called as a progeny, and the progeny ame kihor karone banaiso jate niki we can get both the characters that we want, right? Next, so parents with desired traits are selected and evaluated. Now we have certain methods of selection. Select koriso boli kole etu nohobo, we have to follow certain methods of selection. So what we'll be discussing here is we'll be talking about some common methods of crop improvement, right? So ami ki korilu prothomote collect korilu tarpisot ami parents bilak select korisu, select kora plants bilakor majot we will try to do the cross so that we can get something called as crop improvement, right? So while going for crop improvement these are the points that we have to discuss. Crop improvement or karone kun bilak method apply kora hoy, we go for selection, we go for hybridization, mutation breeding or polyploid breeding. So these are the methods through which the parents are selected and evaluated. The parent to correct ba parent to bhal hoy ne nohoy, offspring to thike ase ne nai, ami jun kita characteristics bisariso are they present in that progeny. So all the studies leads to the study of these 4 points. The first point among them is selection. What is selection? Selecting something is called as selection. Talking about selection, we need to remember that it is the oldest method of crop improvement, right? Agor loke aba aji loke ami jimanbur crops paisu, these are nothing but the result of selection. Aru ei selection bilak kune korise? Prehistoric people bilake korise. Aji je ami bhat tu khai asu ba aji je ami any form of food that we are eating, kiman dinor ba kiman generations before people have been eating that rice, but there should be someone who has selected that rice. No, there should be the first person who told that this is something to be eaten, right? Etu bostu ba etu variety of rice khabo paru, etu variety of mango ami khabo paru, ei bostu tu kune koisile? Kisuman prehistoric people e koisile, right? So selection is considered to be the oldest method. Second point is almost of the entire food that we have, most of them has been selected by prehistoric people, right? Next is selection is or it can be of 3 types. Tinta type of selection kore, eta hol mass selection, eta hol pure line or inbreed selection, and the other is called as clonal selection. So selection kenake koriba? Mass selection koribo para, pure line selection koribo para. Listen or hearing these words, mass selection hok, pure line hok, clonal hok, these are certain words that we already have an idea about. Mass selection means what? Basic characters ami bisari nasao, we take it in a bulk and then we go for improvement. That is what is called as mass. As far as the next one is concerned, pure line or inbreed. What is a pure line? Pure line ki bostu? Self-pollinated plants are junto progeny ulai, and that progeny is repeated again and again for hybridization, and the product that we obtain is called as a pure line. The point that you have to remember is self-pollinating plants, right? Okay, so selection, mass selection, next we have pure line selection, and next we have clonal selection. Clonal selection has come from the word clone. Clone, kot hunisa? Clones are the product of dash reproduction. Clones are the products of asexual reproduction. So এই তিনটা সিলেকশনৰ মাজত the points that you need to focus is in case of a mass selection, a selection to on the basis of কি কৰে This has been selected on the basis of phenotypic characters. Now there are two words, right? Phenotypic and genotypic. What is the difference? Phenotyping mane ki? What is genotyping? C. Yeah, morphological. And genotypic is something related with a gene, right? Right. So if you do something from gene point of view, that is called as genotypic, and from physical appearance, that is called as phenotypic, right? So A2 methods that we follow karo, mass selection that we follow karo, what we see is, we see the phenotypic characters only. In short, what we need to remember is, any selection that is made only on the basis of phenotypic characters is called as mass selection, right? The next point is, mass selection kunbilak plants are koribo pare. Generally, it can be done in cross pollinated plants, right? Mass selection, we go for cross pollinated. So these are only 2 points that you need to remember. Eneu ki hoy, mass selection is not very common nowadays, no? Ajikali to improve methods of selection ulai gose. Nowadays, instead of following all this process, directly ami jodi eta notun plant banabo lage, what do you go for? You go for process called as mutation, right? Imanbur kori tumi collect koriba, select koriba, thik ase. Again you go for a cross, you wait for the generation, right? Instead of going through all this lengthy process, what do you do is, we directly go for some improved varieties like mutation breeding, right? But jehetu yate ami ki mon rakhibo lagbo 2 points. One, it is based on phenotypic characters. Next, it is done for cross pollinated plant. Next we come to the second one, that is pure line inbreed. Since we have the word pure line, so the first point that we need to remember is obviously self pollinating plants. So enukwa dhoroner selection ami kot korim, self pollinating plants bilakor progeny bilak select koribo lagile, we go for pure line or inbreed selection, right? What we do, a large number of plants are selected from a self-pollinating crop and harvested individually. Self-pollinating crop no, tar progeny ulabo, hey progeny bilak ami ki korim, eta eta ke ami harvest korim. Hey harvest kora ba eta eta ke ami grow korim. Hey grow kora plantor pora, ako tahator jetia progeny ulabo, hey khini ami evaluate korim, and then ultimately we will get something that is called as the best progeny, right? As far as these notes are concerned, it is already given, so you can go through the notes properly. So what we will do is, we will just try to understand. So what we have talked about mass selection, and we have talked of pure line. Pure line or duta word, first is, it is done for self-pollinating plants. Agor to ki asile? Cross-pollinated plants, yate ki asile? Self-pollinating, right? Yeah, after self-pollination, progenies will, yeah, the number of progenies that would be formed, what they will do, we will go for harvesting those progenies. Tamane progeny to lom loi pela will be sowing them in the fields. Bahut bilak seasons grow korar pisot juntu best ami pam, that best progeny is released as a pure line. This pure line selection has been commonly applied in a specific variety of food, that is wheat. Right? Our wheat or at an example they are say Kalyan Sona. So what is Kalyan Sona is nothing but a variety of wheat, right? Next we come to clonal selection. Clonal selection, we know we are talking about clones. Clones ko tahisile asexual reproduction. We also remember vegetative reproduction also, right? So vegetative propagate, yeah. Yeah, it is also a wheat variety. Right? Here what I have written is just one variety of wheat, right? There can be so many varieties of wheat also, so many varieties of rice, right? Okay, so clonal selection on nami ki saro, so clonal selection vegetative propagated plants or karne apply kore, right? And then the selection is made among clones. So vegetative adult plant, this plant will reproduce vegetatively, and as a result, what will be the result of what they will produce? They will produce certain clones, no? Clones produce koribo. And what are clones? Clones ki hoy? Morphologically and genetically, if they are similar, they are called as clones. So we might, and morphologically similar, they are called as clones, right? So A2, 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 all these might be considered as a clone. So amongst this clone, which clone is the best? That type of a selection is called as clonal selection. So clonal selection or duta point, vegetative propagated plants, and the selection is made always among clones. One example of a plant is a variety of potato, that is kufri safed variety of potato. Right? Out of all these varieties, no, iman besi important hoy, important kunbilak hoy, especially the points that we will be discussing during Green Revolution. Green Revolution kami kisman varieties for him. At that time, those points you need to remember. Right? Okay. So this was something called as selection. So we are discussing common methods of crop improvement. Our targeted first method to ki asile selection. Selection could be of 3 types, that is mass selection, then pure line selection and clonal selection. Yeah. Next we go to the second point, that is hybridization. What is hybridization? Hybridization mane ki? Yeah, crossing. Hybridization, right? What is hybridization? It is a cross that is done between two genetically different parents or different plants. And why you do the cross for? আমি যে cross to করিলো কিহৰ কাৰণে করিলো? To obtain certain desirable characters. Right? So what is the point that is important is two genetically different or মাজত আমি কি কৰো? Cross koro, right? And why do we do this? So that we can get a new plant with all the characters that we want. There are certain points about hybridization that you need to remember. The first is, in case of hybridization, what happens is, we try to play with the genetic makeup of the plant, right? Ami etal plant or genetic makeup or logar we try to play. So what is the primary aim of hybridization? Primary aim ki to produce variations. So produce variations, right? Plant A into plant B will give you a plant C, and that plant C would vary or differ from plant A and B, and that is what is called as variations, right? Next is hybridization always produce something called as, we have a term that is called as hybrid vigor. Right? Hybridization purhile, hybrid vigor word to, it comes to our mind. Now what is hybrid vigor? Hybrid vigor ki hoy? Have you heard this term? No. Hybridization is a process that leads to hybrid vigor. What is hybrid vigor? Hybrid vigor says that the recombinant or the progeny is always superior to its parents. Hybridization or karone junto progeny produce hole, that progeny shows hybrid vigor. What is hybrid vigor? The progeny is of a better quality as compared to their parents. This is what is called as hybrid vigor. Buji paiso ne? Right? So you need to remember the definition. What is the primary aim? What does it produce? Or it leads to hybrid vigor. Certain other points is, next point about hybridization. Hybridization does not change the genetic constituents or genetic contents of the organism, right? But it leads to new combination of genes. So hybridization process to পঢ়ি থাকিলে কি কৰিব লাগিব কেটামান পইণ্ট আমি মনত ৰাখিব লাগিব the point that we have written next point is does not change the genetic contents no genetic contents যিমান থাকিব যিমান টা numbers of chromosomes থাকিব সিমানেই থাকিব কিন্তু what they do is they try to change the combination of genes right there are just 2 or 3 points that you need to remember about this next is hybridization could be done between two parents or it could be done between more than two parents right so it was a cross jot niki two parents could be involved or it more than two parents could also be involved jedi two parents are concerned or hybridization process to amongst two parents hoye ase tak ami ki boli kom single cross aru etu case ot ami kom this is nothing but a multiple cross right so this is the point just two more points we need to talk about next is first natural hybridization ami kod dekha pai so hybridization is a process of crossing it is seen in natural conditions also or artificial hybridization ami koribo paro right so the point from entrance point of view is first natural hybridization ami kod dekhisu yeah it is seen in corn or maize right jehetu we have talked about natural hybridization we have to talk about first artificial hybridization also so first man-made hybridization you have to remember the name that is Fairchild mule now looking or hearing this word mule you don't have to think about the animal right since we are talking about botany obviously we are not talking about that mule or the cross between a donkey and a horse this is a name that has been given to a hybridized product or a hybrid and that is between a cross of two parents that is sweet william aru carnation eduta ki hoy these are nothing but two flowering plants right so prothomote hybridization jedia korisile dudal plant select korisile edal hol sweet william aru edal hol carnation tahator juntu product ba progeny form korisil that is called as Fairchild mule and Fairchild mule is considered to be the first artificial hybridization so almost we have discussed the points of hybridization the last point you have to remember is the name of the scientist who was the scientist who has introduced this process in crop improvement so hybridization was introduced for crop improvement by a scientist called as Colruyter and the last point is hybridization obviously ami ki koru maximum of the hybridization is man made and jehetu man made you cannot be saying that all the experiments that we do are always successful amar jetia mon gol tetia ami cross korilu and the best progeny that we obtain this is not like this right so what we need to remember is je jiman ami experiments koru jiman ba hybridization process koru only one among hundreds right out of 100 experiments maybe adult plant we produce korim with all the desired qualities that we want right so it is not as simple as we think je ami golu dutal plant cross korilu cross kora loge loge ami best progeny to pai golu that is not so simple we need to go for repeated process to get a certain product so this is what we have studied till today or we have been talking about common methods of crop improvement aji ami duta complete korilu selection or hybridization in the next class we will be talking about the remaining two right thank you